used in clinical research. So why should we use PCR? It's very sensitive. We can amplify 1 copy to 10 copies of DNA, can detect organisms that cannot be isolated. We have rapid turnaround times. However, there are some disadvantages. They can be technically demanding at times, can be expensive. There's risk of contamination. There's a need for rigid quality control measures. Now we move on to the hottest area of research, real-time PCR. It monitors the accumulation of amplicons as the run proceeds instead of at the completion of the run, that is endpoint. There's many manufacturers that supply real-time machines. Some from this slide shows the different manufacturers. Bio-Rad has iCycler, Roche has the Light Cycler, Thermo Fisher has the Quant Studios, Strategene has the Fluoro Tracker, and so forth. So all these systems has some common aspects. They amplifies and detects in an integrated system, uses fluorescent probes used for constant monitoring, has fast turnaround times. That is, they have rapid cycling times. They have sealed systems with low contamination risks. They can be quantitative assays. The Thermo Fisher systems, it's a micro teeter plate format, can process between 96 and 384 samples in 1 to 2.5 hours. Uses real-time amplification and detection in the same system, can perform quantitative results, uses fluorogenic probes with reporter and quenchers, has tap DNA polymerase with 5' to 3' exonuclease activity. So how do TaqMan probes work? The TaqMan probe principle relies on the 5' to 3' exonuclease activity to cleave a dual label probe during the hybridization to the complementary target sequence of the fluorophore based detection. As in other quantitative PCR method, the resulting fluorescent signal permits quantitative measurements of the accumulation of the products during the exponential stages of the PCR. The Light Cycler is an instrument from Roche. It's a real-time detection system, can be used for quantitative results, uses hybridization probes, can detect 5 targets simultaneously, can be used in 96-well format, and has a runtime between 90 minutes and 120 minutes. It's a sealed system, so it's contamination-free. There's currently a number of different probe formats which are used in real-time PCR, of which uses the fluorescence resonance energy transfer. Probably the most common of these are the TaqMan probes. However, we have settled for using adjacent hybridization probes. Unlike TaqMan probes that have, which only uses a single probe, this format uses 2 probes. The way in which these probes work is that the 2 probes align on specific PCR products. This allows the energy from the fluorophore on the first probe to be transferred to the fluorophore on the second probe. It is the energy transfer subsequent fluorescence which is monitored by the instrument. Essentially, as the PCR product accumulates, the fluorescence which is emitted by the reaction mixes increases. So in the Light Cycler, this diagram shows the baseline amplification. Here's the stationary phase, and we can see an exponential increase in fluorescence. So what happens in the PCR itself? At 95 degrees, the DNA strands come apart, followed by the annealing stage, where the 2 probes come together and a fluorescent signal is given off. And this gives us a fluorescent signal, which is monitored by the real-time software. As you can see, the fluorescent signal increases over time. At 72 degrees, we don't see any fluorescent signal. So the applications of the Light Cycler, it's for the detection of infectious disease agents, target characterization, determining of microbial load, that is quantitation. An example, 266 swabs from multiple sites were collected in VTM for HSV culture. 23% were culture positive, confirmed by antigen detection. 113 or 42% were Light Cycler positive following extraction of the VTM using glass fiber extractions. 51 were light cycle positive and culture negative. All these were confirmed as HSV by sequencing. So how does Roche characterize HSV? They have common primers for HSV1 and 2, or pan primers, and they have probes that have different melting temperatures. So we can distinguish HSV1 and HSV2 based on the melting temperatures. As we can see from this diagram, the melting temperature of HSV2 was about 67 degrees, and HSV1 was about 73 degrees. So we can tell the two HSV apart. So the light cycle can also be used for microbial load testing. For commensals, organisms determine a normal microbial load. Elevated levels determine infection. To detect active infection by increasing microbial load, to detect antiviral drug resistance, example in CMV or HSV. So how does this quantitative assay work? We have standards that is serially diluted. As we can see, our standard at 100 million copies comes out at cycle about 2025, and our standard at 10 copies comes out about cycle 40. And we can see that and as we can see, the stronger the standard, the earlier it amplifies. So the advantages of real-time PCR: it has rapid cycling times, it is high sample throughput, over 200 samples a day, low contamination risk as it is in sealed systems, very sensitive, can detect about 3 picograms to 1 genome of DNA, has a broad dynamic range of 10 copies to 100,000 copies, reproducible, allows for quantitation of results. However, it is software driven. PCR detection: the TaqMan and light cycle utilizes probes. Number of chromophores is limited. Alternative technologies are needed. The use of an alternative method is molecular beacons. So how does molecular beacons work? Molecular beacons have a stem loop structure, and has a stem loop structure, as a molecule a stem structure, and a reporter on the 5' and 3', and a reporter and a quencher. These stem loops can be designed to have different melting temperatures, so we can detect multiple targets in the same channel. To detection of amplification of products at the endpoint, multicolor beacons can detect multiple targets in the same channel. Better detection of single point mutations can be used for drug resistance analysis. The next part of the talk will be on the film arrays. It's easy to use, 2 minutes of hands-on time. Results is in under 1 hour. Can detect 42 viral or bacterial pathogens per test. It's contamination free, and contamination is not an issue. Increased sensitivity and specificity. So how do these film arrays work? So you'll insert the pouch, add the hybridization solution, add your sample to the hybridization solution, and insert the pouch into the biofire or the film array. So our sample extraction works in the first part, and then there's 2 phases of multiplex PCRs. The first phase, there's multiplex PCR, and then there's a second nested multiplex PCR. In the first phase, a multiplex PCR is performed, the samples are diluted, and all in a closed system, and a second multiplex PCR is performed, nested multiplex PCR is performed. Biomuria has multiple panels. They have a respiratory panel, they have a blood culture panel, they have a GI panel, a meningitis panel, and so forth. An example: a 69-year-old male patient presents at the ER at 9:30 with fever, cough, and breathing difficulty. Significant medical history with 10 years of worsening COPD, mild kidney failure, temperature of 102 degrees above normal, white blood cell count, elevated PCT, decreased breath sounds in both lungs, low PO2, and new infiltrates on the chest X-ray. Patient is admitted with a diagnosis of bacterial pneumonia, started in the ER on antibiotics, vancomycin, and piperacillin, and tazobactam. The traditional diagnostic would have been to culture the specimens. On day one, a sputum culture would have been taken and gram stain would have been done. A moderate grab on day two, gram-negative rods and numerous Staph aureus, negative, vancomycin de-escalated to cefazolin. Susceptible results, Klebsiella, resistant to PIP and Tazo, meropenem, no certainty and resistant mechanism, switch to ceftazim and avibactam. Now, if we performed a pneumonia panel, we could have detected on day one, Staph aureus susceptible to methicillin, Klebsiella resistant to KPC detected, vancomycin de-escalated 34 hours earlier, PIP and Tazo changed to ceftazidime and avibactam active for KPCs 58 hours earlier. Seegene is another manufacturer that supplies multiplex PCR kits for detection of respiratory infections, GI infections. They have a women's health panel. They have 28 targets in the women's health panel, 25 targets in the GI panel. They have respiratory infection panels of 26 targets. In the STI panels, they have 28 pathogens. And in the meningitis panels, they have 18 pathogens. The in vivo diagnostics of the health status inside the body is usually by X-rays and CT scans. In the in vitro diagnostics, analyzes specimens derived from the body, such as fluid, urine, etc. We have a symptomatic diagnostics, empirical diagnosis. We can have cultures. This is long inspection times, restricted to types of examinations. Immunoassays, unavailable in the window period. Molecular diagnostics, early diagnostics, more accurate than our cultures. The process is as follows. We'll collect a sample, extract DNA or RNA, perform a PCR, and perform the real-time PCR. So workflow, extract the sample. We can use an automated extractor or manual extractor, perform the real-time on the CFX, and use the Seegene viewer. So normal real-time PCR, and we can detect one target per channel. So we can detect 4 targets in a 4-channel system. Seegene uses tag oligonucleotide cleavage extension, where we can detect up to 5 targets in one channel. So we can detect about 20 targets in one PCR. And they're just as sensitive as other singleplex assays. So applications of real-time PCR can be used for screening, for blood donors, preemptive screening for post-transplants, component screening algorithms can be used for diagnosis, viral identification, classification, characterization, presence of active infection prior to serological conversion, can be used for treatment decisions, baseline viral loads can be performed, co-infections can be seen, candidate preemptive therapy can be used for drug resistance to monitor virological responses to treatment, patient treatment adherence, viral suppression, response to guided therapy, resistance emergence, can be also used for treatment of cure, treatment efficacy, viremic resolution, sustained virological response. Real-time PCR is used most commonly in viral loads and has most clinical significance. This diagram shows the suppressed and non-suppressed viral loads in our patient population in Africa. As we can see from the diagrams, the unsuppressed or non-suppressed viral loads ranges between 10 to 20%. So how does the screening work? We have target viral load monitoring, suspected clinical immunological failure, the routine viral load, early detection of virological failure, test with viral loads, greater than 1,000 copies, evaluate adherence issues, repeat viral load 3 to 6 months later, less than 1,000 copies, maintain first line, greater than 1,000 copies, we'll switch to second line therapy. There are many manufacturers that supply viral load systems. Abbott can detect about 40 copies, bioMérieux about 25 copies. Roche about 20 copies, and 13.2 under Cobus. How did we use real-time PCR for our research purposes? Diagnosis of bacterial vaginosis in a resource-poor limiting setting relies on semi-quantitative microscopy algorithms such as nugget scoring. We evaluated by real-time assays to detect and quantify BV-associated bacterial communities. So for our method section, a total of 267 women from Durban, South Africa, presenting with symptoms of STIs were enrolled into the CAPRISA 083 study. Exclusion criteria for the study were pregnancy, HIV positive status, a recent antibiotic treatment, and being a sex worker. Women consenting to the study completed the sexual risk questionnaire. A total of 247 samples were available for analysis. The study was approved by BREC. Gram stain microscopy was performed on vaginal smears, followed by nugget scoring. Specimens are characterized according to scores as either normal, 0 to 3, that was non-BV, intermediate, 4 to 6, abnormal, 7 to 10. Molecular analysis was as follows. We evaluated the presence and concentration of lactobacilli species, Lactobacilli crispatus, Lactobacilli jensei, Lactobacilli innis, and 5 BV-associated microbes, Gardnerella, Prevotella, BV-associated bacteria, Megasphere, Atobium, using the Tachman Thermo Fisher assay. Sensitivities and specificities were generated for each qPCR assay. We see that the sensitivities for Lactobacilli jensei was between 93% and had a specificity of 42%. For Lactobacilli crispatus was 89% and specificity of 37%. For BV-associated bacteria, it had a sensitivity of 72% and a specificity of 75%. Gardnerella had a sensitivity of 78% and a specificity of 79%. We next performed a linear regression analysis on our real-time outputs. And we see that our BV-associated bacteria has a positive association with BV status, and our Lactobacilli species has a negative association with BV status. An increasing in Lactobacilli with a decreasing in BV status. With a decrease in Lactobacilli, an increase in BV status, and an increase in BV-associated bacteria, an increase in BV-associated. When we combined our analysis, we showed that an increase in our BV score is seen as we combine our BV-associated bacteria. We next performed, we next looked at sensitivities and specificities of these Tachman assays and compared them to the gold standards. And we showed that our Tachman assays had a 91% specificity, sensitivity, and 100% specificity. For chlamydia, it had a 94% sensitivity and 99% specificity. For TVAG, it had 100% sensitivity and a 99% specificity. For MT-Intelium, it has a 91% sensitivity and a 98% specificity. Advantages of real-time in a clinical setting. It has a fast turnaround time compared to traditional methods such as cultures and other assays, can identify the cause of disease from a small amount of sample, can perform multiple tests from the same sample, can be used to monitor the disease cause, such as measuring viral loads, can monitor therapy adherence, is a culture-independent method, can be expensive at times. The disadvantages, it can be expensive at times, can only detect what is being looked for compared to other methods such as culture. The primers and probes are designed on a small sequence, and mutations in the sequence may lead to false negative results. Since it needs only a small amount of sample to amplify, can detect organisms that may not be clinically significant, does not tell you if you have an infection-causing disease, as you are amplifying DNA, and this is used as a proxy of infection. Future perspective. Future commercial products of such detection systems are already available, even though not many, such as the BioFi, exist for detection of bacteria and viruses on a chip. More microfluidic PCR-based systems will be developed and commercialized. The miniaturization of the PCRs will continue with the development of new systems integrating sample preparation with qPCR, moving towards a truly portable system to answer systems for POC applications. Thank you.